Hello students, welcome to Scientific online biology classes. So this is the third chapter, that is the human reproduction and the second day. Yes, in last class we studied about the initial introduction. Yes, what is the reproduction, what is the puberty, and what is the reproductive system. Here, so puberty means it is a time or the duration where the organism attains the sexual maturity. From that point onwards only they have the capacity to produce anyone. Yes, so this attainment of puberty is completely the hormonal neuroendocrine regulation is there. So the brain, the hypothalamus will secrete the GnRH will affect the pituitary gland will produce the LH and FSH. These LH and FSH will act on the primary sex organ, that is a testis and ovary. From the testis, testosterone androgen is produced, that will decide the secondary sexual character of the male, and estrogen and progesterone secreted from the ovary, that will decide the secondary sexual character of the female. Yes, so sexual dimorphism also we are seeing. Yes, how the male is completely different from the female one when they attain the maturity. Yes, so next we come to the male reproductive part. In that one only scrotum part we cover. Scrotum means this one. Yes, so this is the male reproductive part, this is a sectional view, and this is the same male reproductive part system one from the front view. Yeah, so in last class we covered this figure. So this is the scrotum. It is a fibromuscular sac which is present from the lower abdominal wall, clear, which is present in the groin area in between the thighs, clear. So this is a sac-like structure. The wall of the scrotum is made up of three layers. The outer, the wrinkled skin, just below that one, cutaneous connective tissue is there. Inside, smooth musculature. Here some special muscles we are seeing. The dartos muscle, yes, one type. So that is present in the scrotal wall. The testis is initially, this testis is present in the abdominal cavity during the fetal development. Yes. So from the 7th month onwards, this testis will descend and enter into the scrotal side through the inguinal canal. This canal is the inguinal canal. So which is packed by the connective tissue. If this inguinal canal is not properly packed or some damage occurred, then the intestinal loop will enter inside that one, scrotum. That will create the inguinal hernia. Yes, we already discussed this one. So this is the scrotal side. The testis is in the suspended state in the form, with the help of the spermatic cord. The spermatic cord present in this region only. This spermatic cord contains the connective tissue where the arteries are there, veins are there, lymphatic vessels and nerves are there, plus vasa deferens and this one, cremaster muscles. So two types of muscles we have. One is the dartos muscle, second one is the cremaster muscle. So these muscles will function and they will decide the shape of the scrotum and position of the scrotum. By doing this, so they regulate the temperature. That's why scrotum is called as a temperature regulator organ of the male reproductive system. Yes, why means? So for the spermatogenesis, 35 degrees Celsius is necessary, but the body temperature is 37 degrees Celsius. So that's why scrotum is localized into the scrotal cell. Yes, so the scrotum is divided into two chambers due to presence of the septum scroti, which is present in the center. Clear? The externally it is visible in the form of raphe. Yes. So this is about the scrotum. Means in some organism, for example, in seasonal breeders, the testis is present inside the abdomen. During the breeding season only, it will descend and come into the scrotum. For example, when you take the otter and the bat. Yes. In some organism, this testis always present inside the abdominal cavity. For example, elephant here. Next, in kangaroos, this scrotum is present in front of the penis bone. So in last class we studied about only the scrotum. Yes. So in this figure means what are the major part means this is the testis. From this testis inside the accessory ducts are there. So this will lead to the epididymis then the vasa deferens will ascend into the abdominal cavity through the inguinal canal. So this is the vasa deferens and it will join with the duct of the seminal vesicle. These green color structures are the seminal vesicle. So after that one, when it joins the ducts of the seminal vesicle, then it enters into the prostate gland. So again it receives the duct from the ureter. This is the duct from the ureter, this is the common duct. So collectively it from the urethra, from this point onwards it is called the common urinogenital duct. So this is the urethra pass through the prostate gland, then just beside this one bulbourethral glands are there. This urethra pass through the penis and open outside the orifice. So this is the male reproductive system. Again, in this class we are going to target this figure. Here, I am going to rub this figure 1, then we will cover the other parts in this figure. So this is about the male reproductive system. Yes, student, we are going to cover the next topic, that is the testis. So in last class we covered the scrotum. So next topic is the testis. So in plural it is called as the testis. Yes. So the testis is, it is a pinkish organ measuring about 5 cm length, 3 cm thickness, 2 cm width, and 12 gram in weight. Yes. So this testis is suspended to the scrotal sac with the help of the spermatic cord, and which is situated over the fibrous tissue that is called the juvenile. So this is the juvenile column. Yes. So this is inguinal canal, and which is held by the inguinal canal cremastal muscles also. So the testis it is covered by 3 layers. So outer tunica vaginalis, which is hollow in nature and filled with the coelomic fluid here. So second layer, middle layer, tunica albuginea. So this is the fibrous in nature. Yes. Third layer, inner, tunica vasculosa. It is a connective in nature, connective tissue. It is a connective tissue. So albuginea is a fibrous tissue. Yes. So now this tunica albuginea will produce the sap, means it will enter into the testis. So in this manner. So this is the testis and this is the tunica vasculosa, tunica vaginalis. So inside tunica albuginea is there. So this is the tunica albuginea, and this will produce the septum, which will enter into the testis and produce the chambers. So this is the tunica albuginea. The black color is the tunica vaginalis. This blue color is the tunica albuginea, which is a fibrous nature. Inside, which is lined by the connective tissue, that is the tunica vasculosa. So these layers are the tunica vasculosa. Limestone testis, the simple testis, is divided to almost 200 to 300 small chamber-like structure. This is nothing but the testicular lobules. So the tunica albuginea, the second layer, enter into testis and produce septum and form 200 to 300 testicular lobules. So this outer layer is a tunica vaginalis. This blue color layer is a tunica albuginea. This green color layer is a tunica vesculosa. Like this one, testis is divided to 200 to 300 compartment, that is called testicular lobules chamber. And each testicular lobule, each testicular lobule have 1 to 4 seminiferous tubules. So this is the functional unit of the testis. So this is the seminiferous tubule. So seminiferous tubule means here almost 1 to 4 seminiferous tubules, like this one. Seminiferous tubules are present. Yes. So this red color is the seminiferous tubule which are present. Seminiferous tubule. Therefore, almost 900 to 1000 seminiferous tubules present in each testis. So gradually, this is the testis. Testis is covered by the 3 layers. In that one, vascular, tunica vaginalis, tunica albuginea, tunica vesculosa is there. Tunica albuginea will produce the septum. Due to that one, compartments are created. These compartments are nothing but the seminiferous, means testicular lobules. In each testicular lobule, 1 to 4 seminiferous tubule is there. And ultimately, each testis consists of almost 900 to 1000 seminiferous tubule. So these are the seminiferous tubule where actually spermatogenesis, spermiogenesis takes place. Yeah. So this means this is the main part. Yes. When we take the seminiferous tubule, we take the sectional view. We draw here. So seminiferous tubule means, so this is the testicular lobule. Yes. So in that one, so these are the seminiferous tubule. 1, 2, 3. I will take 3 seminiferous tubule. Yes. So this is the testicular means lobule. In that one, these are the seminiferous tubule. So seminiferous tubule, the space present in between that one, this is the interstitial space, and seminiferous tubule. Here, so the space present in between this one, these are the space, and where the connective tissue is present. In that one, the lymphatic vessel, arteries, vein, everything is there. So some special cells are present in this space which is present in between the seminiferous tubule. These cells are nothing but the Leydig cell. Yes, so these are the Leydig cell. Leydig cell. So the connective tissue present between testicular lobules have special cells called lytic cell. So this is discovered by the German scientist, that is a lytic, and these lytic cells are these cells produce androgen such as testosterone. So this produce androgen or the hormone like testosterone. So these are the lytic cell. Functionally it is very important. So this is these cells present outside the testicular lobules. So these are the seminiferous tubule here. So these are the testicular lobules. The space which is present between the seminiferous tubule. So where the connective tissue is there, where the lytic cell is present. So these cells produce the androgen or the testosterone. Yes. So inside the seminiferous tubule. Now this is the seminiferous tubule which is covered by the thin membrane, lamina propria. So just downside the basement membrane is there. Yes. Inside germinal epithelium. So this germinal epithelium is made up of some special cells. In that one, two types of cells are there. So this is a germinal epithelium. So in that, this black color cells are called the primary germ cells, and in between that one, some elongated cells are present. So these cells are called the Sertoli cells or the sustentacular cell or the nerve cell. So this is the one seminiferous tubule. Inside it is covered by the lamina propria. Inside germinal epithelium is present. Germinal epithelium is made up of two types of cells. So this black color cell is called as a primary germ cell, and this red color cell is called as a Sertoli cell. So now we are going to study this Sertoli cell and the primary germ cell. Is this clear? So this is about only the testis, testicular lobule and seminiferous tubule. Now we are going to study what are the Sertoli cell or the primary germ cells in detail. Is this clear? Now, seminiferous tubules, yes, covered with lamina propria, yes, germinal epithelium or seminiferous epithelium is made up of two types of cells. Yes, two types of cells. Germinal epithelium made up of two types of cells. One is called a primary germ cell, or it is also called a spermatogonium. Second one is the important cell, that is the Sertoli cell, also called as the testicular cell, or it is also called as the nurse cell. So Sertoli cell, testicular cell, or the nurse cell, and spermatozoa. Yes. Now this is the one cell in testicle. Yes. So it is covered by the lamina propria inside. So these are the Sertoli cell. Yes. So these are the primary germ cells which produce spermatozoa. So these are the primary germ cells or spermatozoa. Yeah. So now I will take only one this one. Yes, spermatozoa undergo division and produce the many cells. These are the primary spermatocytes. Yes, some of the primary spermatocytes will enlarge. Yes, this undergo meiosis and produce the secondary spermatocyte. Secondary spermatocyte again divide and produces 4 spermatids. Yeah, so these spermatids will release and attach to this Sertoli cell and develop into spermatozoa. The complete process we are going to cover in the spermatogenesis process one. So in this one, so the germinal epithelium is made up of 2 types of cell: primary germ cell, and Sertoli cell. So these are the germ cells which undergo meiosis and produce the primary spermatocyte, secondary spermatocyte, and the spermatids. These spermatids will attach to the nurse cell. So Sertoli cell means, so this red color cell is functionally very, very important. So it is called nurse cell and testicular cell because so these cells are little bit elongated and non-dividable. These are under the influence of the pituitary hormone. Yes. So what is the main function of Sertoli cell means? It provides the function of Sertoli cell means it provides nourishment for the developing spermatozoa, provide nourishment means even exchange of the gases and the exchange of the nutrient take place through this Sertoli cell. When we take this here, just beside this one, primary spermatocyte, secondary spermatocyte, spermatids are attached one. So it provides the nourishment for the developing spermatozoa. First one, that's why it is called a nurse cell. Yes. So second one, this Sertoli cell produce the main protein, that is the androgen binding protein. So androgen binding protein means, so these are the protein. This is the androgen binding protein or ABP. So this produces the androgen binding protein. So the androgen or the testosterone, which is produced from the Leydig cell, come and attach to this androgen binding protein. Then concentration of the testosterone increases. So this produces the androgen binding protein. Third one, if the testosterone or the concentration of the FSH increases one, then this nerve cell also produces one protein, that is called the inhibin. Yes. So this will create the negative feedback. If the excess amount of testosterone or the pituitary hormones are secreted, this will go and it will stimulate the pituitary hormone to stop the production of the pituitary hormone. So it produces inhibin also. It produces so the function of inhibin will study the regulation, hormonal regulation of the spermatogenesis one. So ultimately, due to providing the nourishment. This is called the nerve cell. It produces the androgen binding protein and it produces the inhibin also. So Sertoli cell is functionally very important. Here, so due to its particular shape, the Sertoli cell maintains the shape of the seminiferous tubule. So this is about the internal structure of the seminiferous tubule, where the primary germ cell, which undergoes spermatogenesis, spermiogenesis and produces spermatozoa. Nerve cell means or the Sertoli cell or the testicular cell, which are present inside, and that will maintain the shape of the seminiferous tubule. It provides the nourishment for the developing spermatozoa. It produces the androgen binding protein and it produces the one more hormone, that is inhibin. So this will produce a negative feedback. So this is about internal structure of this seminiferous tubule. So once the spermatozoa produced inside the seminiferous tubule, these are the spermatozoa produced by the spermatogenesis and spermiogenesis. So it must transport it. Clear? So now the function of the accessory ducts and the accessory glands. So this is a function of the testis, where the spermatogenesis takes place and the testosterone is produced from the Leydig cell wall, and the androgen binding protein and inhibin produced by the nerve cell or the Sertoli cell or the testicular cell. So this Sertoli cell is discovered by, so this is discovered by Enrico Sertoli. Yes. So this is about the testis. Now come to the accessory duct and accessory gland. Now what happens? So spermatozoa produced. Now try to locate which are the accessory duct present in the male reproductive part. Again we will write the flow chart. So this is the seminiferous tubule, red color. Yes, these are the seminiferous tubule. Almost 1 to 4 seminiferous tubule present in the east testicular tubule. Spermatogenesis takes place inside the seminiferous tubule. This spermatozoa is released into the irregular network of the structure that is called the rete testis. This blue color structure is a rete testis. Yes. So which is present on the outer side. Here. So this rete testis collected means joined to form almost 10 to 12, 10 to 20 tubular structure. So this green color structure is a vasa efferential. Yes. So first one is a rete testis, means seminiferous tubule. Initially means one side it is a straight structure, they are called the tubulary. Yeah. So gradually they became the irregular structure, network of the tubular structure, that is called the rete testis. Rete testis collectively form almost vasa efferentia. So this vasa efferentia, almost 10 to 20 vasa efferentia collected, and they open into the one more complex structure, that is the epididymis. So this is the epididymis. Yes. So epididymis is little bit complex structure. Almost the length of this epididymis is almost 4 to 6 meter. Yes, but it is highly coiled and highly packed. So this epididymis again divided to 3 parts. Yes. So when you observe this one, this is the head region, this is the middle region, this is the talar region. The epididymis is again divided into 3 parts. So that is capute epididymis, corpus epididymis, and cauda epididymis. Capute means head. So this is the capute. This is the head region. This is the capute. And corpus means it is the main body. This is the corpus region, central region. And cauda means it is the talar region. It is present in the lower side. So capute epididymis, corpus epididymis, and the cauda epididymis. So length is almost 4 to 6 meter one. Whatever the spermatozoa produced inside the seminiferous will first feed into the radiate testis, then it enter into the vasa efferentia, then the vasa efferentia, 10 to 20 vasa efferentia collectively dump the spermatozoa into the epididymis. Here, so in complete observe this structure, this radiate testis, vasa efferentia, and this one, these structure means ultimately this tubuli recti. So I got this one. Now the topic is accessory duct. Accessory ducts. So first one is the tubuli recti. It lead into rete testis. So this is the rete testis, irregular structure. Then rete testis open into the vasa efferentia. Efferentia. Yes. This vasa efferentia open into little bit longer structure, that is a epididymis. So epididymis is almost 4 to 6 meter. So which is divided into caput, corpus, and cauda epididymis. This is caput epididymis. This is corpus epididymis. This is cauda epididymis. So the epididymis is present outside the testis. So these three ducts, rete testis, this tubuli recti, rete testis, and this one, these are collectively called as the intratesticular ducts. Why is all these three are present inside the testis? Yes. So this is the testis, the rete testis, vasa efferentia, and tubuli recti. All are present inside the testis. That's why it is called the intratesticular accessory duct. Now, epididymis is the outer part, almost 4 to 5 meter. So this is a highly coiled structure, caput, corpus, and cauda. The epididymis open to the next structure, that is the vasa deferens. So epididymis open into vas deferens. Yes. So this coiled structure. This is the vas deferens. So this is the vas deferens. Yes. So now vasa deferens is almost 35 to 40 centimeter. It is a 35 to 40 centimeter long. Initially it is a coiled structure. Later on it became straight and it passed through the inguinal canal and enter into the abdominal cavity. Yes. Now this is the extra testicle. Now here testis plus otis epididymis. Epididymis is collectively called as, means this one and this one collectively called as testicles. Only this structure. Testis plus epididymis is called as the testicles, and the ducts which are present inside the testis, these are called intratesticular duct. So testicles means that is nothing but the testis plus epididymis, not the vasa deferens. Now vasa deferens will enter into the abdominal cavity, almost 30 to 40 centimeter long. It will come near the almost ureter one, will encircle the ureter, and it joined with the duct of the seminal vesicle. These green color structure of the seminal vesicle. So this is the duct of seminal vesicle. So this is the vasa deferens and form the common tube here. So it joined the duct of this one and it entered into prostate network. So these green color structures are seminal vesicle. Yes. Just before the joining of the duct of seminal vesicle, the vasa deferens will dilate and it is called as the and pinna. Yes. So ultimately, whatever the spermatogenesis are produced inside the seminiferous tubule, either they store into the radial testis, vasa efferentia, and epididymis. So vasa efferens is the conducting part. So inside this epididymis, the serostratified epithelium layers are there. In that one, some primary cells are produced. So they produce some glycoprotein. So this glycoprotein will provide nourishment for the developing spermatozoa. Still the spermatozoa are inactive. So now this is the antenna from the both sides. This is the vasa efferens, this is another vasa efferens, and they dilate and produce the ampulla, and they join the duct from the seminal vesicle, and they enter into the prostate gland. This red color gland is a prostate gland. Prostate gland. Before that one, this blue color structure. So this is a duct from the ureter. So this is a ureter. Means urinary bladder is also present here. From this urinary bladder, this duct comes and joins this common duct. Now from this point onwards, this duct is a common urinogenital duct. So from this one, the fluid from the seminal vesicle will come. From this one, urine will come. Sperms, and from this one urine will come. So this is a common urinary duct. From this point onwards, it is called as the urethra. After vasa deferens, the next part is called as the urethra. Yes, from this point onwards up till here. Yes. Now urethra is again divided into four parts. The urethra is divided into four parts. Which one? First one is the urinal urethra, through which only urine passes. Next one is the prostatic urethra. So this is the urethra will enter into the prostate gland, that is called the prostate urethra. Next one is the membranous urethra. So this is a membranous urethra where the bulbar urethral gland will open into this one. Last one is the penile urethra. So this is the last part where the urethra passes through the penis, which is a muscular copulatory organ. So ultimately, when you take the accessory duct, in that one, tubular recti is the part of seminiferous tubule which are straight in nature. Radial testis is the highly irregular structure. This one. Vasa efferentia, these are the structures which are leaving the testis one and enter open into the epididymis. So almost it is the longest part, 4 to 5 meters. So again, it is divided to caput, corpus, and cauda epididymis one. Epididymis is open to vasa deferens. This is the tubular structure which originates from the caudal epididymis one, and its initially this coil structure. It is almost 35 to 40 centimeters long, which enters the abdominal cavity through the inguinal canal. So it joins the duct from the seminal vesicle and also duct from the urinary bladder and from the common urinogenital, that is called the urethra. This urethra is again divided into urinal urethra, then prostatic urethra, the membranous urethra, and penile urethra. Yes. So this is about the accessory duct. Yes. What is the function of accessory duct means? So it is a conducting part. So in some cases the spermatozoa is stored. Till the ejaculation occurs, the spermatozoa is stored here only. Once the ejaculation occurs, the spermatozoa is loaded into the female reproductive duct. So the main function is the storing, maturation, and the conduction of the spermatozoa from the tubuli recti up to the urethra. So this urethra is a common urinogenital duct which are present outside. When you compare this one up to the vasa efferens, these are the intratesticular duct. Epididymis, vasa deferens, and urethra, these are the extratesticular duct which are present outside this one. When you combine this testis plus epididymis, then it is collectively called as the testicles. Yes. So this is about the accessory duct. So we covered only tubular part. Still the accessory glands are there, where the seminal vesicle, prostate gland, and bulbourethral gland. This is a bulbourethral gland. This is bulbourethral gland. Now we are going to cover the three parts, that is seminal vesicle, prostate gland, and bulbourethral gland. So what they secrete, how they are functionally important, now we are going to do. Yes? Next one is the accessory glands. In that one, first gland is the seminal vesicle. So these are paired in pair. Second one is the prostate gland. Third one is the corpus gland or the bulbourethral gland. So just want to be covered the accessory ducts. In that one, tubular rete testis, vasa efferentia, epididymis, vasa deferens, then urethra we covered one. So this is accessory ducts one. Now collectively these three are the accessory glands. First one is the seminal vesicle. Vesicle. Yeah. So it is musculoglandular sac. Musculoglandular sac, almost measuring 5 cm in length. Yes, when it is a condensate, if you uncoil this one, it is almost 10 to 15 cm long. If you uncoil this one, yeah. So it is a musculoglandular structure. Usual length is a 5 cm. If you uncoil that to 10 to 15 cm, yeah. So when you compare the complete composition of semen, it produces 70% of semen made up of seminal fluid. Yes. Remaining is the secretion of the prostate gland, bulbourethral gland, and even the accessory ducts. 70% of the semen is made up of seminal fluid. So the secretion is thin, little bit viscous, mucoid, and alkaline in nature. Yes, little bit alkaline in nature. The secretion, it secretes main, that is the fructose and prostaglandins. Fructose will act as a nutrition purpose for the nutrition of spermatozoa. Yes, prostaglandin will promote the uterine contraction. In female reproduction, it will increase the uterine contraction. Yes, it will increase the uterine contraction. So only two components are there. Fructose, which will provide the nourishment for the developing spermatozoa, for the nourishment for the spermatozoa. Prostaglandin, which will increase the uterine contraction, so that the spermatozoa will ascend into the uterus. So this is the seminal vesicle. Clear? Only two parts are there. Next one is the second one, prostate gland. Seminal vesicle means they are paired. Two are. This is one, this is another. Second one is the prostate gland. Prostate gland. Prostate gland means this is a red color structure. This is a larger one. So this is the tubulo-alveolar gland. Tubulo-alveolar. So here almost 30 to 40 small tubulo-alveolar glands are there. They will open into this urethra, separate independently, and secretion of this one almost it is measuring about 10 to 15 cm, a little bit larger. Yes. So this will produce 20 to 25% of the semen. 70% of the semen come from the seminal fluid. 20 to 25%, almost up to the 30% come from the secretion from the prostate gland. So its secretion is thin, milky, and slightly alkaline. So this is also alkaline, this is also alkaline. So this is little bit thin, no, this is a thick. So secretion from the seminal vesicle, thicker one, here it is a thinner one. Yes. So what they secrete? The secretion of prostate gland, it contains calcium, phosphate, citrate, profibrinolysin, thromboplastin, and cephalin. Apart from this, so this will produce the major enzyme, that is fibrinogenase and acid phosphatase. The secretion of the prostate gland contains the fibrinogenase and the acid phosphatase. The concentration of this will decide the activity of the prostate gland. Yes? The removal of the prostate gland, the major point. So, main point, removal of prostate lead to sterility. Yes. So if you remove this prostate gland, then it leads into the sterility of the male one. So that's why functionally prostate gland is very, very important. So last one is the corpus gland, or the third one. Corpus gland or bulbourethral gland. Yes, seminal vesicles are in pair, they are 2 in number. Prostate gland is only 1. Yes, when you take the bulbourethral gland or the corpus gland, they are also pair in number. Yes, pair. So this is also tubulo-alveolar gland, but the secretion is not that much important. But so the secretion is called as a precum. This is a precum. One is, so spermatozoa produced here, seminal fluid produced here, and prostate fluid is produced here. So before they enter into urethra, the secretion of the bulbourethral gland will lead first. Here, the secretion will first means the secretion of the bulbourethral gland first secreted one. Why means, so this is alkaline in nature. So this will increase the lubrication also. And this will neutralize the acidic path of the urethra. So if the urine is passed, that is a little bit acidic. So this will neutralize the acidity. Yes. So that's why it is called the precum. So this is about the accessory gland, seminal vesicle, prostate gland, and the bulbourethral gland. When you collectively call, so secretion of these 3 glands and the spermatozoa, that is called as semen. Yes. Almost the semen is consisting of the secretion of the seminal vesicle, secretion of the prostate gland, secretion of the bulbourethral gland plus spermatozoa. So the pH of the semen is almost 7.3 to 7.5. Yes. So what it contains means it contains everything. It contains fibrinogenase, then profibrinolysin, fructose it contains, prostaglandin it contains. So semen is a fluid which comes from the male reproductive part, which contains spermatozoa and secretion from these 3 glands. The pH is 7.3 to 7.5. So this is about the accessory ducts. Now last one, the external genitalia. External genitalia means So all these organs are present inside one. Externally only scrotum is visible, and this the male copulatory organ, that is the penis, is visible. Yeah. So this is the penis. Yes, it is the last part one. So it is copulatory organ. It is erectile in nature. So through which urine also pass and the semen is also pass one. So when it is the transverse section of the penis, this is the penis. So this is the penis. So when it is the transverse section of this one, so in that one, the transverse section, so highly erectile the tissues are there. Yes. So these three columns of tissues are there. So it is covered by the common muscle connective tissues. So these are the erectile, means shaft-like structures are there. So this is called the carpora cavernosa. This is a carpora spongiosa. So this sinusoid-like structure. So if the blood flow into this one, this became erect. Yeah. So these are called the carpora cavernosa. So upper two structure, and the lower one is called the carpora spongiosa. Yes. I will write. The space is not there. I will draw. So this is the transverse section of penis. So upper to this structure. So this is the corpora cavernosa, and lower one, this is the corpora spongiosa. Yes. So total three structures are there. This will extend from the anterior part to the posterior part. The carpora spongiosa, this is the urethra. Yes. So these are the musculature. So whenever the organs occur, means this the penis will attain, means it will erect, and that will facilitate the insemination function. Here. So this is the penis one. The anterior part is little bit dilated one. This is called the gland penis. So this is the gland penis, and it is covered by the foreskin. Yes, or profuse. Yeah. So this is the gland penis. It is by the skin one, that is called the foreskin. So this is the external genitalia which is externally visible. So the penis is erectile in nature, that help in the insemination, through which the urine also pass and the semen also pass one. So this is all about the male reproductive system. So here only complexity means accessory duct and accessory glands. Yeah. So this is the today's class. Again we will meet in the next class where we are going to cover the female reproductive part one. Till then, work hard.